Okay, so we finished the variant cycle. And we're going to talk a little bit about the fertilization and development, pregnancy. So what happens during fertilization? So first of all, as you remember, by the way, that's a mistake right there. It's not ovum. Okay, that's secondary oocyte. Secondary oocyte enters the fallopian tube. Okay, that makes sense. That's the moment, right? In the meantime, as sperm is moving through the vagina and uterus and comes closer to fallopian tubes, it gets capsulated. It's basically a fancy term for it completing its maturation. The prostaglandins and oxytocin produced during the intercourse, and prostaglandins that are in the seminal fluid, they stimulate the contraction of the uterus and contractions of the fallopian tubes, which promote sperm movement towards the oocyte. Usually, the oocyte is fertilized in the fallopian tube. If you think about chances, with every ejaculation, we're talking about tens of millions of sperm cells deposited in the vagina. About 1,000 sperm cells actually reach the fallopian tube, and only one fertilizes, which is another excellent confirmation, one of my favorite theses about nature. Nature is about anything except efficiency. It doesn't have to be efficient. If it works, it works. Okay? Now, in the head of the sperm, if you would look at the structure of the sperm, that's the head, and that's the flagellum. In the head of the sperm cell, the structure called acrosome. Acrosome contains enzymes that dissolve the protective layer surrounding secondary oocyte, called corona radiata. When corona radiata is dissolved, sperm cells can penetrate the membrane, okay, of the oocyte. So sperm binds to actually special receptors on the layer that surrounds the oocyte, so-called zona pellucida. Okay, and that binding initiates the fusion of the membranes of the sperm cell and secondary oocyte. Now, really interesting response that occurs in now fertilized ovum is the release of so-called cortical granules from the cytoplasm of the oocyte. It is stimulated by increased calcium levels. The function of cortical granules is to shut the door behind that sperm cell. Cortical granules make membrane of now fertilized ovum impermeable for sperm cells. Does that make sense? So one sperm cell enters, and that entrance stimulates the release of cortical granules, and cortical granules will make ovum impermeable for any more sperm cells, because during fertilization there's only one sperm cell that can enter, should enter. Does that make sense to you? So at this point, now the question, can such thing happen that two sperm cells enter one oocyte? The answer is yes, it can. It's called polyspermy, and those eggs do not survive. There are protective mechanisms that will basically destroy eggs with polyspermy. Now, when these two cells fuse, you've got what's the word? Secondary oocyte is arrested in the anaphase 2. So everything is lined up. Sister chromatids are lined up on the equator of the cell, but they're not yet separated. So at this point, separation of the spermatids happens, and now you've got mixed genomes. Okay, you've got two haploid genomes from the sperm cell and from the oocyte that, by mixing, form the functional diploid cell, diploid zygote. That makes sense? So this diploid zygote is formed in the ampulla of the fallopian tube, as we mentioned before. Zygote gets transported through the fallopian tube into the uterus over a period of 3 to 5 days. The forces that transport the fertilized egg are the fluid flow, first and foremost. The mucus that is present in the fallopian tubes is propelled by ciliated epithelium. Synchronous beating of the cilia on the surface of the fallopian tubes will move the zygote towards the uterus. Contraction of the fallopian tube will also contribute to the movement. And the reason why transport takes a bit of time is that during that time that zygote spends in the fallopian tube, it divides. And it basically partitions into more and more smaller cells. Here you see that zygote develops into a blastocyst. So here you can see a blastocyst enlarged, okay, with the first traces of amniotic cavity. Does that make sense? So what I want to highlight, it's kind of an interesting thing in embryogenesis, is that when zygote divides, it is division without generating new same-sized cells. It's actually partitioning of a zygote into many smaller cells. So it is separated in two parts, and then each part is separated into two smaller parts, and so on and so forth. So when blastocyst is in the uterus, it implants in the endometrium. Cells that help to implant the blastocyst in the endometrium are called trophoblasts. They release proteolytic enzymes that essentially weaken the endometrial epithelium, allowing blastocyst to attach and incorporate itself into the endometrium. And then elevated levels of progesterone, which, as you remember, will be observed here. This graph represents the progesterone dynamics without fertilization. With fertilization, it would look something like that. Okay, more plateaued. So elevated levels of progesterone will lead to swelling of endometrial cells, which store nutrients, and they become decidual cells, which will provide nutrients. Okay. In the meantime, trophoblasts will establish the communication between the decidual cells and blastocyst. So trophoblasts essentially play the role of placenta. They deliver nutrients to the developing embryo all the way until placenta will be formed. If you will look, at this graph, you will notice that, that's the time of ovulation, and all the way up until week 12, after the last menstruation, week 12 after the ovulation, trophoblasts deliver nutrients to the developing embryo. Only at about 12 weeks, placenta starts to develop. And at the time when placenta develops, it takes over the nutrient transport completely. You see how fast the diffusion across placenta increases during the embryonic and fetal development. Okay? And as placenta grows, essentially what happens, it becomes bigger in terms of the size, surface area increases. But its thickness becomes lower and lower, so its permeability increases as well. Okay? This image, you can see the ovum, the layer of trophoblasts surrounding ovum, and endometrium on the sides. Does that make sense? Now let's talk a little bit about the structure of placenta. And I will say a few words about some placental cells, actually, later. Love that topic. So here, you can see placenta, and you can see villi. So each of these structures is the villus. Okay? That villus is at the very end, so-called trophoblastic cord. Okay? Can you see the villus and the trophoblastic cord? In the villus, you're going to find venules and arterioles, okay, here. And what I want you to understand very clearly is the direction of blood and what blood goes where in the umbilical circulation. So first of all, the fetus is attached to the placenta by the umbilical cord. That makes sense? When umbilical cord is removed postpartum, that's belly button. Clear? Now, let us think, what is the function of placenta in relation to the fetus? What's the function of placenta? Nutrients, and most importantly, oxygen. And oxygen goes to where? From placenta to fetus. Okay. So blood that is oxygenated should go in which direction? Use words placenta and fetus. From placenta to the fetus. Does that make sense? So here you can see that. Now, one more question. Blood vessels that go from placenta to fetus, they're going to be veins or arteries. From placenta to fetus. Veins, exactly. Because they basically go to the heart of the fetus. Does that make sense? So, you have to understand that umbilical veins take the oxygenated blood. Here you can see, well, umbilical vein, one umbilical vein, takes the oxygenated blood, sorry I mixed it up, umbilical vein takes the oxygenated blood to the fetus, while umbilical arteries take deoxygenated blood from placenta, from fetus to the placenta. So in this case, this placental circulation on the fetal side, which circuit does it remind you of? Which circuit? In the adult life. Pulmonary, yes. It's like pulmonary circuit. Umbilical artery is taking deoxygenated blood to the placenta, which plays a role of a lung here. And umbilical vein takes the blood back from the placenta to the fetus, back to the heart. Does that make sense? Do you understand the idea of villus, blood vessels in the villus, and which blood vessels deliver the blood from fetus to the placenta and back? We good? Now, how does this exchange work? Trophoblastic cords and villi are practically bathed in mother's blood. Okay? So if you would look closer at this image, you will notice that each villus is surrounded by what we call a sinusoid. So those are sinusoids. Essentially, these sinusoids are, very permeable capillaries. We can call them spaces. They're not really capillaries. They're like empty spaces that are filled with mother's blood. Does that make sense? Do you understand what I'm saying? So here you can see the uterine artery of a mother. Uterine artery of the mother will deliver the blood into that sinus. Okay? This is called a marginal sinus, by the way. Will deliver the blood into the sinus. Gas exchange will occur between the sinus and the villus. And then deoxygenated blood will be drained by the uterine veins. Does that make sense? So uterine veins take the blood away from the placenta to the mother's heart, and its blood is deoxygenated, while uterine arteries deliver oxygenated blood to the placenta. In a fetus, the situation is different. Umbilical arteries deliver deoxygenated blood to the placenta, while umbilical vein gets oxygenated blood back to the fetus. Does it make sense? In the mother, uterine arteries deliver oxygenated blood to the placenta, and uterine veins take deoxygenated blood from the placenta back to the mother. In the fetus, umbilical arteries take deoxygenated blood to the placenta, while umbilical vein carries oxygenated blood back to the fetus. Did that work? Cool. One more time. Okay. Mother, uterine arteries, oxygenated blood to placenta. Uterine veins, deoxygenated blood from placenta. Cool? Fetus, umbilical arteries, deoxygenated blood to the placenta, umbilical vein, oxygenated blood back to fetus. We good? Okay. Now, sinusoids. Sinusoids. No, that's a bad color. You're not going to see it. Something like dark, really dark. Sinusoid here and here. And blood vessels in the villi, they're separated by the layer of cells that we call trophoblasts. Sometimes they're also called syncytial trophoblasts. Now you can relax for a second. I just love that topic. It's not going to be on the exam, but the story is great. Placenta and fetus, they are basically isolated. mother and fetus are isolated from each other to a large extent by the placenta. That makes sense? Now, several years ago, 2 pregnant women working with, I believe, influenza virus, asked themselves a question. Are we supposed to do that? We're pregnant. And they decided to find out how placenta separates mother and the fetus. Why a lot of infections that mother gets during pregnancy do not affect fetus at all. It was known before that trophoblasts, sometimes they refer to as syncytial trophoblasts, they form tight junctions that prevent any transport across placenta. And it was discovered about a decade ago that the protein syncytin that forms that barrier was actually derived, ironically, from a virus similar to HIV that infected our predecessors, some apes, probably tens of millions of years ago, okay? And they developed what the form of placenta that we have. But those 2 women actually found another mechanism. It turns out trophoblasts produce microparticles that package antiviral proteins that control viruses in the maternal blood that is delivered to the placenta. Which I think, for me, this story is fascinating because women just, they were sitting working and they asked each other, quite literally asked, are we supposed to do that? And from that, the project arose and they published, and it was pretty great discovery. Does that make sense? So trophoblasts essentially isolate mother and fetus from each other. Yes? Small molecules. They sometimes, many of them, they fat soluble. For them, trophoblast is not a problem. So for instance, why there is, what's that, alcohol fetal syndrome? Alcohol is small lipid soluble molecule. It has no boundaries. It will get to the fetus directly. Same way there are infections that actually can trick this trophoblast and cross placenta. Usually, if we talk about small molecules, they can do that. Okay, so, and that's drugs, alcohol. Oh yeah, totally. Formaldehyde would cross placenta. Absolutely, 100%. Yeah, yeah. That would be a terrible idea. Yeah, yeah. You store corpses in formaldehyde. Yeah, that's preserve to preserve. By the way, kind of a side note, if you think about the relationships between the fetus and the mother, it's a typical parasite. And one of the reasons why bloods are isolated, so mother doesn't develop an immune response against the parasite. Actually, mothers, during pregnancy, women are moderately immunosuppressed. If they wouldn't be, they would reject, most likely, that parasite. Okay. Now, one thing that I want to focus your attention on, and one thing that I'm going to be asking, important, is the mechanism of oxygen transport from mother's blood to the fetal blood. Okay? So, fetal blood that enters through the umbilical arteries into the villi carries carbon dioxide. Is that clear? That's what arteries, umbilical arteries, are carrying. So this carbon dioxide will diffuse into the maternal blood. Now the question is, how does this diffusion, how does elevated level of carbon dioxide in the maternal blood, how does it affect pH of maternal blood? Lower, exactly. Okay. In maternal blood, it becomes lower. And carbon dioxide leaving fetal blood, how does it affect pH of the fetal blood? Increases. Now it's time to remember what's the Bohr effect is. In the Bohr effect, decreased pH increases unloading of oxygen from hemoglobin. Now, decreased pH is in the maternal blood. So hemoglobin in the maternal blood starts to unload oxygen. Elevated pH is in the fetal blood. So hemoglobin in the fetal blood starts to load oxygen. This is called double Bohr effect because pH changes in both maternal and fetal blood. Does that make sense? Now that represents the concentration of oxyhemoglobin in the fetal and maternal blood. You can see that red line representing the hemoglobin dissociation curve of the fetal blood is shifted to the left, which clearly represents elevated loading, and maternal elevated unloading. On top of that, fetal hemoglobin has higher affinity to oxygen. Does that make sense? Altogether, it makes sure that the concentration of oxygen and hemoglobin is higher in the fetus. Okay, that everything is saturated as much as it can be. You clear? That's really important. Now, what diffuses where? Basically, maternal blood receives carbon dioxide and wastes. You see the main, so what are the main wastes? Fetus, does it have metabolism? Sure does, right? Speaking of metabolism, what are the two products of oxidative respiration? CO2 and water. So CO2 goes into the maternal blood. Water, it's water. Okay? Now, not only this, fetus also metabolizes proteins. Products of the protein metabolism are different compounds of like urea, uric acid. They go to maternal blood, bless you. Okay? Creatine, product of creatinine phosphate metabolism goes into the maternal blood as well. That makes sense because, think about this, kidneys don't really work, they don't really pee, technically speaking. So all that cleanup happens in the placenta instead of the kidneys. Did you get double Bohr effect? I'm going to ask about it. Now, some hormones. I'm going to tell you what you need to know, what you don't really have to focus on. First, HCG. Now, what the acronym HCG stands for? Human chorionic gonadotropin. So this is the hormone that is secreted by syncytial trophoblasts. Remember, we talked about this. You see how trophoblasts reach a maximum function at about week 6. This graph, you can see that chorionic gonadotropin that is produced by trophoblasts reaches at maximum at around week 8, slightly after. When you use a pregnancy test, the strip-based, electronic, it doesn't matter, what essentially is detected is the chorionic gonadotropin in the urine. Does that make sense? when you see advertisements, now this test is like, allows you to detect 1 week earlier, we're basically talking about sensitivity. The concept, I just learned about it second year of chemical school. How these tests are all like principle. It's chromatography test. The concept is pretty simple. It's been known for, I don't know, decades. It's just how well you can detect it, and stuff like that. The functions of chorionic gonadotropin is basically to maintain corpus luteum. Corpus luteum means estrogen and progesterone. It also stimulates the growth of endometrium and prevents menstruation. So trophoblast, think about this. If there are menses, there's any problems with the blood flow, typical blood flow response in endometrium, then implanted blastocyst will be washed away with menses. And that happens. That may happen. Okay. Also, not all, if somebody has multiple, what should I call it? Multi-embryonic pregnancy, like triplets or twins or quadruplets, not all blastocysts that got implanted will actually develop. We were told that we actually have 3 in the beginning. We ended up with 2. So one blastocyst just never developed. Was implanted but got stuck. Okay, something. Now, that's one hormone that you need to know. Another hormone, progesterone. I'm going to go in a certain order. So progesterone is critical for preparing endometrium for implantation and to prevent uterine contractility in the beginning. If uterus will contract, blastocyst is gone. It's going to be expelled. That makes sense. We'll also provide some nutrition for morally developing blastocyst, and it will prepare breasts. Now you can see that progesterone starts to decline closer to the end of pregnancy, because if you think about that, before actual delivery, before labor, uterine contractions should not be inhibited. They actually should be stimulated. So that's estrogen. Estrogen will not only initiate uterine contractions at the very end during labor, which we'll talk about, but will also stimulate the growth of uterus, which is absolutely necessary, as you can imagine. As the fetus becomes bigger and bigger, uterus has to become larger and larger. It will also increase the breast size and the size of external genitalia. Okay. Now, other hormones change, like adrenocorticotropic and thyroid stimulating. Adrenocorticotropic will recruit the energy storage from the fat deposits, and thyroid stimulating hormone will stimulate protein synthesis in the mother. Gonadotropins will decrease expectedly. One hormone that sometimes does not receive enough attention is somatomamotropin. Now, if you think about the name, somatomamotropin. Mamo means breasts, somato body, so breast body, tropin where it goes to. Somatomamotropin stimulates breast development. Okay. It also has a very interesting effect on the glucose metabolism by mother. Glucose uptake in the mother goes down, which makes more glucose available to the fetus. I want to be very clear. You need to know about 4 hormones: chorionic gonadotropin, estrogen, progesterone, and somatomamotropin. For these 3, you need to roughly know the dynamic. What I mean by that, if I ask you at the end of pregnancy, Which of the following hormones is practically non-existent? You tell me. Chorionic gonadotropin. Am I clear? So basically you have to know this graph with these 3 lines. Did I make myself clear? So which hormone? I'm not going to ask you which levels are higher, estrogen or progesterone, but you have to know that estrogen and progesterone are much higher than HCG. Good? No questions? Now, we were talking about fetus mostly. Let's talk about mom. Pregnant woman will gain some weight, okay, due to the increased size of breasts, increased size of uterus, of course, fetus. So if you would look at this graph, fetus contributes about a quarter, okay? About 1/3 is accumulation of fat. And 14%, 1/7, is increased volume of fluid in the system, like blood, increased volume of blood, increased volume of extracellular fluid. Graph in the right upper corner shows you the volume of maternal blood, and you see that it dramatically increases in the last trimester of pregnancy. See that? Okay. Now, not only this, BMR increases, which makes perfect sense because woman has to produce nutrients for the developing fetus. Nutritional requirements increase and sometimes change. And what's important, blood volume increases and cardiac output increases. Now, if you think about this, that's quite a drastic change for a woman's body. Elevated blood volume leads to elevated blood pressure, right? So that may lead, but not necessarily leads to a condition known as preeclampsia. In preeclampsia, blood pressure dramatically changes. It becomes dangerously high, and that affects kidneys, leading to proteinuria, appearance of protein in the urine. Okay, that leads to improper filtration in the glomeruli, which leads to retention of water and retention of sodium chloride. Does that make sense? Now that leads to further spasms, decreased glomerular filtration rate, okay, and decreased glomerular filtration rate leads to accumulation of toxins. Does it make sense? Preeclampsia is, I don't know how to say, clinical, it warranties early delivery, the C-section, okay? Even if it happens fairly early in the pregnancy, like if it's 6-7 months post-conception, it's going to be preeclampsia, can be life-threatening because it can develop into eclampsia. So at this point, fetus will be delivered by a C-section. Eclampsia is basically an extreme version of preeclampsia. So vasospasms, which sends the blood pressure absolutely through the roof, may lead to seizures. Kidneys practically shut down. The shutdown of kidneys leads to toxemia, presence of toxins and wastes in the blood, which may eventually lead to coma. Think about this. If kidneys are shutting down, there is no proper balancing of pH in the blood, and blood becomes acidic. Acidosis almost inevitably leads to coma. So usually preeclampsia is diagnosed timely. In this case, mother's health usually is taken into consideration first. So if it's relatively late in pregnancy, then fetus can be delivered by C-section. We now, I believe now the cutoff, there were examples of successfully nurturing fetuses that were born at the weight of 1 pound. But that's kind of an extreme thing. So that's really, really small fetuses. Okay. Does that make sense? So you need to know the basic idea of preeclampsia, elevated blood pressure, kidney damage, decreased GFR, and eclampsia as being an extreme version that leads to toxemia. Labor and delivery. Some contractions are pretty normal during pregnancy. I always feel absolutely awkward when I talk about it, I'll be honest. Because like if I talk about cardiovascular system, respiratory system, you know, I have it, you have it, we all have it, fine. But when I talk about especially delivery, I feel absolutely terribly awkward. So I have no idea. I really have no idea what I'm talking about. Okay. So you probably know more than I. Not probably, you do. Okay, will. So weak and slow contractions are called Braxton-Hicks contractions. And they change into the labor contraction during the labor. So what happens from the physiological standpoint? That thing I feel more comfortable about. Okay? Like hormonal regulation, that I feel more comfortable about. Remember at the end of this graph, progesterone goes down, while estrogen keeps getting elevated. So that elevated estrogen leads to increased contractility of the uterus. Estrogen essentially initiates contractions. Okay? When contractions are initiated, baby's head starts to push on the cervix of the uterus. That pressure is sensed by mechanoreceptors. Mechanoreceptors send the feedback to posterior pituitary to release more oxytocin. Oxytocin directly stimulates uterine contractility and also stimulates release of prostaglandins from placenta, which will stimulate uterine contractility. So that elevated uterine contractility forces baby out, and baby's head pushes on the uterine cervix even more, sending signal back, increasing prostaglandins and oxytocin, and that's the positive feedback loop. That makes sense? Okay? So baby is eventually pushed out. Okay? Baby is pushed out, no pressure, oxytocin is not released anymore, contractions basically cease. Make sense? Once the baby is out, it's delivered, umbilical cord is cut, placenta detaches and is removed. Okay. Good? Now, you know that there is a medical advice immediately after delivery, and we're just talking about initial delivery now, but immediately after delivery, baby should be breastfed. Reason for it, when baby suckles on the nipples, it sends two stimuli. First stimulus, it's a mechanical stimulus of a nipple that goes to hypothalamus. Hypothalamus releases prolactin releasing factor. Prolactin releasing factor acts on the anterior pituitary. Anterior pituitary releases prolactin. Prolactin increases milk production. That makes sense? That's the half of the deal. Because while mammary glands will be producing milk, it still has to be moved through the lactiferous ducts to the nipples. The hormone that is responsible for that movement is oxytocin. So suckling will also elevate the release of oxytocin from the posterior pituitary. Does that make sense? Oxytocin will reach breasts and will stimulate smooth muscles that surround lactiferous glands to contract, and that's called let down reflex. Milk will be ejected, basically it's like peristalsis sort of, will be ejected from the nipples. Does that make sense? So that initial suckling stimulates both milk production and milk ejection. And that regular feeding the baby will stimulate milk production in a woman and ejection of milk. That makes sense? Any questions? That's also the topic that I don't know much about. Oh yeah, if you're curious which animal milk is closest to the human milk, I can tell you. Whale. Yes. Whale. Whale milk is, by composition, is the closest to the human milk. But it's a bit hard to milk them, as you can imagine. So there was an idea, actually. There was an idea to develop whale farms, but I believe it's a bit cost inefficient.